Probably many of you have heard that there are problems with honeybees in the United States, so I'm going to characterize my talk by giving you a brief overview of what's happening in the United States. And this seems somewhat relative because if you read my title, it's addressing the sustainability of beekeeping in the 21st century. I'm not sure what my title actually is in the program, something like research and extension or something. Does that sound about right? So the deal is I'm going to tell you the problem and then tell you how I've developed a program in Florida to address that problem. And I know you say to yourself, why is Florida relevant to me? Well, I'm hoping something that I say to you this morning is relevant to your own particular beekeeping situation. Yes, it's sunny and hot in Florida all the time, unlike I'm sure what you get most of the time. But I'm sure that there's something in here that hopefully you will find interesting. Of course, I want to thank Mazemore Apiaries as well. I hope I got the right logo. I found that on the internet last night. So I think that must be right. And so we'll start from the beginning. How many of you are beekeepers? How many of you don't have any bees at all? So you're wannabes, right? You want to be beekeepers. Well, I know that I'm preaching to the choir this morning, but I just wanted to share with you. In the United States, we have a U.S. federal agency called NASS, the National Ag Statistics Service, and they keep up with statistics on all things agriculture in the United States. If you want to know the number of sheep in California or blueberries in Maine, etc., all of that has been captured and posted to the NASS website. So using those data, we know that in 2013, because every year they report last year's information, in 2013, based on the report that came out this year, we had around 149 million pounds of honey produced by about 2.64 million honeybee colonies in the U.S., and the value was somewhere around $283 million. So that sounds like a lot, right? We all think of honey when we think of honeybees. We think honey is extremely important. But in reality, in the United States, honey is almost a nuisance to us because even though the vast majority of beekeepers are hobbyists and keep bees to make honey, the vast majority of colonies are owned by commercial beekeepers who use those colonies to pollinate crops, usually. So most beekeepers you run across in the U.S. will tell you about all the honey they produce, but most colonies in the U.S. are used to pollinate crops. Make sense? So when compared to the value of pollination, we suspect there's around 15 billion US dollars added to the pollination of our fruits, vegetables, nuts, and berries, and increase in crops every year due to honeybees. So you could argue, rightly so, that honey is really a nuisance compared to pollination. For us, we keep bees to pollinate crops. In fact, about 1.7 million colonies, roughly 60% of all the colonies that we have in the United States, go every year to California to pollinate almonds. Just almonds. And the U.S. is a big country, right? It's a few thousand miles, I guess kilometers as well, from one side to the other. And so we constantly truck bees all over the country. For example, in Florida, which is about as far away as you can get from California, a lot of our beekeepers go to California first to pollinate almonds in February, and then go to Maine or North Dakota or Florida, etc. The average commercial beekeeper who pollinates crops goes to 4 different crops with their colonies, moving them around the country to help with fruit and vegetable set. So pollination is a really big deal to us. And out of that estimate, if you read the reports, somewhere in the neighborhood of the report suggests about 1 in 3 bites of food, but just kind of conservatively looking at it, about 10 to 20% of the food production come from the efforts of honeybees, we estimate in the U.S. So that's somewhere between 1 out of every 10 bites of food to 1 out of every 5 bites of food. And that's a lot. You could argue, probably rightly so, that honeybees are the most important ag commodity in the U.S. because 20% of what I eat is not dairy, it's not poultry, it's not wheat, but it is due to honeybees. So you can see what we do as beekeepers is very important to the world, right? You know that already, I'm sure, but I just want to reemphasize. Probably, though, many of you have heard that honeybee populations in the U.S. are declining, and that same statistics service, NASS, has been keeping up with the number of honeybee colonies since the late 30s until today, and we peaked at nearly 6 million honeybee colonies in the late 40s, and it's been a steady decline ever since. So you could argue in 2006, when we had this massive honeybee die-off and the world's dying and we're all going to starve to death, that was actually not a new thing. We've had a problem with sustainability of beekeeping in our country for a very long time. In fact, in 2006, when that colony collapse disorder was first discussed in the U.S. literature, we have plateaued, if not gone up just a little bit in the number of honeybee colonies in the U.S. Of course, if you read the reports, it's the worst thing on the planet. We're losing 35% of our colonies every year. But you know beekeepers split colonies to recover those losses. So we're going to do a simple early morning exercise to illustrate this. We're going to just use 30%. If you have 100 colonies and you lose 30% over winter, how many colonies do you have? 70. If next year you lose 70%, how many colonies do you have? 49. Very good. So you get my drift. We keep going like that since 2006, and we have how many colonies left in the U.S.? We shouldn't have hardly any. You know, physicists tell us if you drop something and it bounces halfway up every time, it never stops, right? Because there's always half of something to keep going to, right? So I'm not saying we'd ever zero out if we lost 30% of our colonies every year, but we would have a lot fewer colonies than we do now. And that illustrates the point that we're losing a lot of colonies, but we're also recovering those colonies. So what you read about in the reports are gross losses. They're not net losses. And net losses were around 1 and 2% every year, which is what this line illustrates, which you could still argue is not sustainable. You don't have to be an amazing statistician to follow this out and realize that we bottom out somewhere over here. So we still need to develop some sort of programming to address this issue that's happening to our bees. Actually, they're formerly your bees, but they're now in the U.S. I don't want to pretend to lay ownership of the bees that you so graciously gave to us a few hundred years ago. Now, so the question is does anyone care? And if you've ever seen me give lectures in the U.S., and I know at least one person has, I like to illustrate this idea like so. I would argue that every bee scientist with a brain and a pulse has been on the news about why bees are dying. We have all been interviewed. CNN, Good Morning America is one of our kind of famous, notable morning news shows. But I like to show you the public opinion view of things like this. If you Google the word honeybee, you get about 36.4 million hits. Okay, I did this last night, so it's accurate. There's one catch. You have to Google it the way the Americans spell it, which is 2 words, and Google it the way you spell it, which is 1 word, and then you have to add the hits together. Incidentally, there is a right way to spell honeybee, and so you'll learn about it tomorrow. If you come to my lecture tomorrow. So if you Google it and add it together, you get about 36.4 million hits. Just to show you how brilliant the public is, if you Google Lady Gaga, you'll get around 221 million hits. If you Google the English Premier League, which I hope is a real thing, I looked it up last night to see if it was real. It seems to be real. Around 99 million hits, and just for the sake of being fair, if you Google William and Kate, you get about 219 million hits. This illustrates a very important point. Popular things are about 3 to, let's just say, 6 times more important than surviving. Now, how many of you are familiar with American politics? Americans aren't either, so don't worry. But we have something called an electoral college. Do you understand have you ever heard about that? The popular vote doesn't actually get the president in office. It's the electoral college. It took me forever to figure out why that's the case. Some of you are going to get that in a minute. Some of you won't ever get it, possibly, but that's okay too. You have a different political system. All right, so before I talk to you about what we do to address these sustainability issues, I wanted to just tell you some of the things that people are looking at. We don't talk about colony collapse disorder anymore in the States. We talk about colony losses. That's more accurate, and it allows us to study anything that kills bees. And here's a list of things that people routinely go back to when they're considering what may be harming honeybees. So again, I'm not going to speak about any of these things in detail. I just wanted to show you some possible culprits contributing to colony losses. I will show you some things that have been popularized but likely are not contributing to honeybee colony losses in the U.S. Let me just throw out the quick caveat and disclaimer. I work for the state of Florida. The University of Florida is a state institution, so technically I'm a government employee, I suppose. So I just want to say, as a scientist and a government employee, I am willing to believe anything as long as there's data to support it. So if we find out that cell phones are killing honeybees, I'll be the first to believe it. It's okay with me. I'm just throwing out there that we don't believe they are at the moment. Besides, cell phones don't even pollinate honeybees, or honeybees don't pollinate cell phones. I just blew my joke. They do, though. They do pollinate blackberries. So, oh, come on. You've got to do better than that. I can't help it. I mean, I'm doing the best I can. You're a Thursday morning crowd. Anyway. All right, moving on. You actually can kill a honeybee with a cell phone. All right, locating bee colonies too close to power lines, unusual solar activity, shift in their Earth's whatever field, the rapture, alien abduction. Again, I would gladly believe any of these as long as there are data to support it. All right, so with that introduction to get you to where I am, the University of Florida is what we call a land-grant institution. Every state should have a land-grant institution. And land-grant institutions are generally, originally, 100 years ago, based on agriculture, because that's what people needed to know about 100 years ago. They were all ag-related problems. So land-grant institutions typically have the strong ag programs and are usually where the bee researchers are, because bee research is tied to ag. And so land-grant institutions will always beat into your head that they do three things. One of those is instruct. When I tell people I'm a professor, the first thing they ask me is, what do you teach? Because in the U.S., that's what they believe professors do, because most people interact with professors from the classroom, not from the standpoint that they do anything else, right? Research, which you know, and extension. Extension's a little difficult to explain outside the U.S. Some people call it outreach or service outside of the U.S. But if you wonder what extension is, I'm doing it right now. Basically, I'm taking data, digesting it, and feeding it to a clientele group in an effort to try to help. Right? So videos, training documents, websites, lectures, workshops, all of that stuff's extension. So I actually have appointments in all three of those areas and have to develop programming in each of those to address sustainability of bees and beekeeping in the U.S. So I'm going to spend a lot of time talking to you about the research we do in my lab, and just a teeny little bit of time at the end talking to you about some of the things that we do here, again, to address this overall issue of bee problems that we have. So generally speaking, there are three areas of research that we do in my lab. The first of those being honeybee husbandry. If you're wondering what husbandry means, it just simply means we do research to try to figure out how to keep bees better, to make it easier to keep bees. Second of those is integrated crop pollination. I'm going to briefly mention that. That doesn't appeal to a lot of beekeepers usually. And then the third of those categories is honeybee ecology and conservation. I will tell you that most of our disease and pest control and bee nutrition work and pesticide work goes in that first category. And that first category is what I'm going to spend a lot of time in this morning talking to you about. All right, so within this paradigm, for example, honeybee husbandry, beekeepers still say in the U.S. that their biggest problem is Varoa. Varoa mites get you three ways. First way that they get your bees is, I need all of you to do this. I'm the speaker, so you have to listen to me. All right? I need you all to do this. I need you to do this. So far, so good. I need you to do this. If you were a honeybee, that would be a Varoa mite on you. So Varoa mites are really small to the naked eye, but compared to their host, they're one of the largest parasites known on the planet. So Varoa mites get bees three ways. The first of those ways is they're on the outside of the bee sucking their blood. The second way is they give bees pathogens, deformed wing virus, for example. And the third way that Varoa mites get bees, at least in the U.S., is, I believe, since we have to put chemicals in colonies to try to stop them, the chemicals hurt our bees. So sometimes I believe the control is worse than just having the pest in the first place with some of the things that are going into our hives. So they suck bee blood, they give bees pathogens, and then we try to do things that maybe aren't so good to stop them. Of course, you can see a bee here with deformed wing virus that we took a picture of here in my lab some time ago. I have a postdoc, Daniel Schmel, who was very interested in determining how Varoa are resistant to various compounds, and that largely was sparked because of Nico and Gudrun Kerniger. I know, Nico, you're next, right? Yeah. Nico is next. They're colleagues of mine from Germany. You're going to want to see both of their talks. Nico was visiting my lab sometime, I believe it was early last year when he did this, and found evidence to believe, a reason to believe that the Varoa mites that we had in our colonies were resistant to multiple acaricides. Not just one, but perhaps had cross resistance to 2 or 3 of the active ingredients that the beekeepers in the U.S. were using to control them. So out of that idea, Daniel Schmel decided that he was going to try to map Varoa resistance to the pesticides across an area where we live. So he went to various beekeeper operations and he collected bees from their operations, exposed the Varoa on those bees to one of 3 different acaricides to see how Varoa are resistant to these different compounds over this geographic range. Now, I'm not going to spend a lot of time talking about all the details, but I will say that the experiment looked like this. The beekeepers allowed us to come to their colonies and collect 30 packages of bees, each package about 2 kilograms, so they gave us 2 kilograms of bees from each of their colonies. And then that package would receive either amitraz or coumaphos or fluvalinate. And those are the 3 active ingredients that we can legally use in the U.S. Coumaphos in Checkmite Plus, I don't know what you might call it here, even if you have access to it. Fluvalinate is Apistan, and amitraz is called Apivar in the States. So basically, he would expose the bees and the mites to this strip, and then he would change the strip every day. So it's 3 days in a row, a fresh strip to try to get the mites. He was overdoing it to see the level of resistance. I also do some small hive beetle research. My PhD was with small hive beetles. This is an adult beetle, eggs, the larva that emerges, and that pupa. So I'm giving 4 talks, and I believe my 4th talk is titled Honeybee Pest and Diseases, if I'm not mistaken. It's been requested of me to change that to a talk on small hive beetles. I don't know if you know this, but small hive beetles have been found in Europe, in Italy. Well, very likely reproducing populations in Italy, so they asked me to change my disease and pest control talk to a small hive beetle talk so that you guys would kind of be mentally ready for any questions you might have about small hive beetles. So that's all I'm saying about beetles. I'm giving a whole lecture on it I believe it's my 4th lecture. We also spend a lot of time looking at pesticide impacts on beekeepers. A lot of beekeepers in Florida are convinced it's the pesticides that are damaging bees and not really these other things. And so I'll share with you how I'm studying this. Probably all of you have read in books and in magazines, etc., that bees will fly. I tried to be ambidextrous with my distances. I failed miserably in some instances, but bees will fly roughly 2 to 3 miles is what the books say, roughly around 5 kilometers. I have some colleagues who study bee flight path from honeybee colonies and say that you can get them from 5 miles and 8 kilometers away from the nest, which is a statistic that's pretty amazing, right? We always say a honeybee can fly 7, 5, 6, 7 kilometers from the nest. That's amazing. But I've never seen anyone do the area of a circle that has that radius, pi r squared. And if you do the math, a circle with a 3 to 5 mile radius is 28 to 79 square miles of area, or up to 200 kilometers of area that a single honeybee colony could, not necessarily would, but could forage in. Does that not blow your mind? So if you do the quick math, it would only take about 7.7 colonies to cover all of London. So some people say, well, clearly what's happening is bees are just going out there and they're being exposed to all kinds of things and bringing it back to the nest. So some people are saying that, so we're trying to look at that. So there's a PhD student now who's going to refine or develop in vitro rearing protocols for queens and drones. And then you can do a whole assortment of things when you've got queens and drones. If you've got queens, you can look at egg output, longevity, etc. This is going to make Nico and Gudrun happy. When you've got drones, you can do drone flight to DCAs, and are they the drones that make it to DCAs, are they full of the amount of sperm, the control drones, etc. There's all kinds of things you can do. So that's future direction. All right, we'll get to there. That's all in the first category of research we do. The second two won't be as long, so just hang out with me a little bit. We do integrated crop pollination. That's real simple. You know this. Most Americans don't. Honeybees aren't native to North America or South America. Our bees came from here and other places in Europe. We do have one race of honeybee from Africa. Americans call that the killer bee. The Africans call it the bee that they use. We are really good at over-sensationalizing everything. Anyway, this is what we do best. We sensationalize. All right. So in Florida, we have nearly 320 species of native bees. Bumblebees, sweat bees, carpenter bees, etc. So we do some research on looking at the contributions of those bees to the pollination systems of our major crops. So the idea is we're trying to help growers develop the best practices to augment their natural populations of bees by planting Florida native wildflowers and minimizing pesticide use and putting out bee nesting habitat, etc. The idea is you're integrating multiple things to achieve optimum pollination, in addition to using the honeybees we provide. Does that make sense? So I have a postdoc and students heavily investing in this. I mean, I minimize it for you because it's not honeybees, but it's probably a third of what we do. I have a big team working on this around the clock. The third category of research that we do is honeybee ecology and conservation. This is the category that most interests me, that I wish I could spend all of my life doing, but unfortunately I have to do other things. I will tell you that honeybees are not native to North America, so we cannot study wild honeybees. We can study feral honeybees, but we can't study wild honeybees, and you know the difference. Wild implies that they are native to that area and they have no history of domestication. In our case, all the honeybees that are living in the trees in the forest trace their lineage back to someone's white box somewhere in the past. Right? So we have feral bees, not wild bees. I'm very interested in wild bees. Europe is a very difficult place to study wild bees as well, because you have a lot of bees in the wild, but they're probably more likely to be feral than they are wild because of what Varoa mites did. Varoa mites probably wiped out a lot of them, and then you repopulated the wild with the bees you had. Could be wrong. That's debated at the moment. But I'll say, why study bees in a place that's questionable? Instead, go to some place you know there to be wild bees. And so I did my PhD in South Africa, which is why you can hear the South African accent I have. And so it's a joke. So I lived there for 3 years at Rhodes University here, and I will note my supervisor, Randall Hepburn, my PhD supervisor, passed away last week. I believe it was last week, maybe 2 weeks ago. So I should have dedicated this lecture to him, but he really gave me a lot of the skill sets that I have to be able to do the type of work that I do. So I just want to note that I did my PhD there with him. And what you're looking at is just a picture of South Africa. So let me give you some ideas of what I want to do with this ecology and conservation. So I did my PhD at Rhodes University right here. And you'll notice that this green area, any plant people in here? Plant people? All right, good. So there is a plant kingdom in the southern tip of South Africa called the fynbos, F-Y-N-B-O-S. It is the most species-rich, the most biologically diverse plant kingdom on the planet. If you want to see amazing plants, go to the fynbos. So this is its distribution here. And they've got this really neat bee there called the Cape honeybee. And its distribution is this blue line. See any pattern? So up here they have that bee we call the African bee or the killer bee. Down here they have the Cape bee. They just fascinate me. Fascinate me, fascinate me, fascinate me. Again, I just want to talk to you forever, but I've got a time limit, so I have to stop. Long story short, all of you know that your colonies, when they lose their queen and they produce laying workers, your laying workers lay unfertilized eggs, and unfertilized eggs become drones. I said that all with one breath. I think I might pass out now. Cape bees, on the other hand, when they fail to requeen themselves, their workers can lay unfertilized eggs, and their unfertilized eggs become females. Ask me how in another talk. I don't have time. It's amazing. So Cape bees can produce clones of themselves. It's amazing. We need to talk about it later. So out of a worker, you can get more workers or even a queen. So that all happens here. Cape honeybees are a big problem for African beekeepers, beekeepers of African bees up here, because Cape bees can get into their colonies and become social parasites and take over the colonies. I don't have time. You can ask me about it later if you're interested. So in the U.S., they're very anxious about getting Cape bees. They don't want to get them. So they funded me to go over and begin developing really easy ways of identifying Cape bees. The problem is Cape bees are honeybees, Apis mellifera, and they're hard to look at and tell the difference between their bees and our bees. So I went over. This is actually my oldest child. He and I went last year to South Africa to collect samples of Cape bees. I have 4 children. Boy, girl. 6, 4, 3, and 1. And so when they hit 4.5, they're old enough to travel with Dad. So that's my oldest son carrying samples with me. What you can't see is there's baboons in the bushes grunting at us. And the guy who was with us is like, oh, don't worry, you're here. I said, why should I be worried? He's like, well, if you weren't here, they'd take your kid into the bushes and pull him apart. I was like, okay. It's like, okay, I feel better now. I'm glad I'm here. So that's him carrying samples. That's me carrying samples. And I sent back a team this year to collect more samples. And basically what we've done is each of those dots represents 3 apiaries from 3 different beekeepers whose colonies, with 20 colonies per apiary that we sampled. That was a mouthful. Basically, we brought back 1,100 some odd colonies worth of 30 bees per colony to my lab. They're all in a freezer. So what are we going to do with those? Well, we're trying to develop ways to identify them easily. It's hard to tell the difference between these girls and these girls, if there is a difference. More on that later. If you invite me back in 3 years, just hang on. But basically, I have a student who's going to try to use molecular techniques to be able to separate the 2 populations. And I have a student who's going to use morphometric techniques to separate the populations. Here's an example. Probably many of you know classical morphometrics to separate the races is based on measurement of various body parts. Again, I have to tell you, you guys are in the presence of some amazing bee scientists in this room, Nico and Gudrun Kerniger. They're technically academic children of Rutner who did all kinds of work here. In the South, in the United States, we would say you're in high cotton, which means you're in the presence of some very distinguished people. But their supervisor and others did a lot of work with morphometrics of honeybees. Well, wings, for example, have a geometry associated with them. There are veins on the wings, and they intersect at 19 different places. So in theory, you can map the XY coordinates of all of those spots, right? If this is an x-axis and this is a y-axis, number 6 may be over 3, up 4, right? You can map the XY coordinates of this and see if there are patterns in these things across the races. So we're doing classical morphometrics to separate the populations, and then genetics and wing analysis to see if we can use one of those techniques to tell us the same thing the classics do. Why would this be useful? Well, if, for example, we could do this, we're currently in the process of writing a computer program that can take a picture of this and find those points. Once you figure out the math, the computer program should be able to just take the picture, find the points, and tell you what bee it is. So our long-term goal, maybe highfalutin, is to develop an app that we can use to take pictures of bees' wings in the field and it tell us best guess what population it believes we're working with. I know that's big, but if you don't aim big, you're going to just be regular, right? All right, so why is this useful to me? Why is this useful to me? Well, it's useful for a few reasons. Remember, I want to do honeybee ecology and conservation. That's all about keeping a bee out of the U.S., but what I really do is I have a secret second mission. I want to be able to be in the field and know what I'm working with on the spot without having to go back and do all these crazy dissections and then only sometimes getting it right. And that's useful to me because I'm interested in these conservation topics. My wife did her master's in South Africa. She worked, it's common for South Africans who have farms to sell their game or to convert their farms where they raise sheep into game farms because they make a lot more money allowing people to come down and shoot their animals than they do eat the sheep. So what they do is in South Africa is you've got farms, 5-year-old game reserves, 10-year-old game reserves, 20-year-old game reserves, and always been game reserves. And you get this nice continuum to look at honeybee populations and ask questions like, when the land's reclaimed, does it benefit bees? How long does it have, how far into the future? You know, all these conservation type questions. So I had a master's student, for example, go around and find all kinds of wild honeybee colonies in South Africa. There's a sheet of propolis in a cliff. The bees are going in those holes, and they built a propolis wall to use as their wall. And once you do this and you map the distribution of honeybee colonies in the out in the wild, you can begin using GIS technology to ask some really cool questions. You guys have a minute for me to explain this? All right. How many of you are familiar with GIS technology? Don't worry, most people aren't, but I will explain. It's real simple. Every spot on the planet has layers of information associated with it. For example, if I were to step outside, there would be an elevation, a rainfall, a climate, a taxing zone, etc. All kinds of layers of information associated with that spot. So when you find honeybee colonies out there, you can use those layers of information. This is fictitious. I just got this off of the web. It's not even honeybees. I just wanted to show you an example. You can map honeybee colonies and see areas where they are most dense, the highest densities, and you can begin asking questions like, why do bees nest where they do? Is it related to rainfall or this or that or the other, etc.? And then you can begin asking and answering conservation level questions. And then we can begin using that technology and those ideas to figure out, where should we keep our bees? Where are our pest populations going to be lowest? Things like that. So it really does have an end purpose and an end goal, but it all starts with being able to identify the bee quickly. We do some work with African bees or Africanized bees in the U.S. That's it for the research. That was one leg of the stool. But don't worry, I know I've only got 5 minutes left. The other 2 legs I'm going to do very quickly. There's 3 legs of the stool, you just got 1. The research is what I'm most passionate about, can you tell? But I will tell you, I've only given about a 20% appointment research, so that's what I'm supposed to do 20% of my time. But all kinds of things have come out from our lab. I've been fortunate enough to collaborate with some colleagues to edit books on research methods related to honeybees. The Kernigers and I, who will be speaking next, the Kernigers put together this book on the mating biology of honeybees. How many of you want to read about bee sex? I had to say it quietly. My daughter, don't say that word ever again. There's a whole book that's coming out that they wrote that is absolutely fascinating. It will blow your mind. It's great nighttime reading. I don't know why that makes me laugh. I should progress. All right. Extension. Very briefly, the extension. So most of what I'm supposed to do is extension. So the good news is I have a big lab. So, 20, 25 people at any given time. Postdocs, graduate students, technicians. They go out and train beekeepers. I train beekeepers. I talk to beekeepers. I'm speaking here 4 times and 3 times in the rest of the country over the next week and a half. My point being is I travel a lot and talk. We do training videos and newsletters and websites and all kinds of stuff that we put out to try to help beekeepers based on the research that's coming out. We do very big events like Bee College, which is where we train beekeepers. Our Bee College in the U.S. is so popular, we started a South Florida Bee College. So we have a North Florida Bee College and a South Florida Bee College. Last year we started a Caribbean Bee College. So we've done that 2 years in a row. So every year we do those 3 training events for beekeepers. If you want to come see bees in the Caribbean, that'd be a great opportunity. We run a Master Beekeeper program. We do all kinds of training. I could spend days talking about this. I just wanted to minimize it so I could spend more time on the research. I'll stop with that part there. I also teach students. I have graduate students, PhD students, and master students. Our department, the entomology department at the University of Florida, is extremely big on distance education. So almost all the courses that we offer face-to-face, we also offer online. In fact, my beekeeping course is exclusively online. If you come to the University of Florida to take beekeeping, you have to take it online. You can take this beekeeping course online. Anybody on the planet can take this course. But it's exclusively online. But that's all in that instruction branch of the stool. So we teach students, masters, PhDs, and undergraduates. We do extension and we do research. Of course, when I say we, you almost exclusively know the boss doesn't do this stuff anymore. We're kind of the figurehead of the program, the CEO, as it were. So if I took a picture of our lab every month, it still wouldn't be enough to keep it up to date, because we constantly are adding new people and getting other people into jobs. So I think about 6 or 8 months ago, this was it, and we still had about half of the team missing there. So I just want to make a point to thank, my team members. I won't go one by one, but they're really the ones who do all the work. Thank you, of course, for your attention. Let me give you my email address, because I have a whole big website. We're revising it now that's chock full of information related to bees. I'd be happy to point you there, but let me get you there through the email address. It's easy, hopefully, for you to remember. J.D. Ellis. My middle name is Douglas. That's where the D comes from. J.D. Ellis at UFL, which should be intuitive because that's University of Florida, .edu, which stands for education. J.D. Ellis at UFL.edu. I sincerely appreciate your attention. I may have a minute for questions. I don't know where we are. Not a minute?
When you had the reduction in hives, did you have a similar reduction in honey production at the early slide you showed? You showed like the quite dramatic reduction.
Oh, so those colonies okay, that's a good question. So the question was when we had the reduction in hives, do we have the same reduction in honey production? It appears to be the case. So I'm going to expand on that. It's really, that's a great question. It's unfortunately, sometimes when I show that graph, a lot of people will look at it and say, aha, that proves that something started killing bees, because a lot of people will point out the late 40s is when pesticides gained in popularity, and as they went up, bees went down. The problem is correlation is not causation. I could correlate that curve with the number of McDonald's in the United States. Right. So my point is, that's a great question. Honey production has gone down. It is possible that we're just needing fewer colonies to achieve the same purposes. Maybe we have stronger colonies than we've ever had. Fewer, but stronger. Maybe the shift towards pollination has done that. But you're right, honey production has gone down. One more question and then I'll turn it over. I know I said 3, but I lied. Well, I'll do 3 since I see 2 hands. Both of you, I'll be nice.
Great. Extension is about reaching out to farmers, but it's also a two-way conversation.
Sure.
I wondered how you worked out the priorities from beekeepers. How do you decide
Fantastic
what to work
question.
on?
Question was, extension is 2 ways. It's not just from me to you, it's also from you to me. How do I prioritize what I do? Well, first of all, I go around to a lot of bee meetings and listen to beekeepers. In Florida, they talk about one thing nearly exclusively: pesticide impacts on bees. Secondly, they talk about Varoa mite impacts on bees, which is why I do a lot of effort in those 2 categories. But with extension, it's not just research, it's also how to deliver the information appropriately. Because some people like face-to-face, some people would rather have it on the pilot. So we do surveys of our beekeepers, asking them, what's important to you and how can I get you information appropriately? So they told me in research surveys, we think this is, and so hobbyists, commercial, and sideliner beekeepers rank their priorities, and I develop research projects that address the priorities for those 3 categories, and then I'll look down at the question that said, once I get these data, how can I get them to you? And I get it to the beekeepers the way that they ask for it. And on top of all of that, I always do extension evaluations to make sure that the information is getting there, that it's being retained, and that it's changing their life. And if it's not, I adjust my extension delivery methods to address that deficiency. Great question. One more question and we shall conclude with my talk.
I was going to ask you about the synergistic effect of pesticides.
Oh, great question.
Are you doing any work to that effect where the effect of 2 pesticides would have greater effect than each
Great question.
individual?
The question was, what about the synergistic impacts of pesticides? Am I doing any work? All right. So I hope what this talk has illustrated to you is that I have a very small brain. All right? And my brain just cannot cross 2 pesticides. And I'll tell you why. There is one bee scientist who's looking at the interactions of pesticides at Ohio State University. His name's Reed Johnson. The problem that I have with synergistic interactions is it gets messy immediately, because you can have these doses of pesticide A and these doses of pesticide B, and then you have to do all the combinations, and before long the study gets too big and too complicated. So what I will tell you, the reason your question is brilliant is because honeybees don't live in plastic containers in an incubator. They live in a colony that might have 4 or 5 residues, let's say 5 residues, and Varoa, and small hive beetles, and deformed wing virus, and nutritional deficiencies, and. So there's a lot of interactions that are going on. And so the reason that I focus mainly on the singles is because it's the easiest, cleanest thing to do at this point. And to be frank, I'm scared to go to interactions before we know what these things alone do? And I'm going to just throw out another spin-off of that great question, in that when I talked about Roundup, what did I really keep saying to you? Glyphosate. Well, glyphosate's the active ingredient. If you look at a Roundup bottle, glyphosate might be half to 1 to 2% of the physical product. The rest of the product is inert ingredients. At least that's what it's called in the U.S. Is it inert ingredients here, other compounds? And there's reason to believe in some instances that those other compounds, the surfactants, the adjuvants, etc., that were put in that product to help it do its job better, might be toxic or synergize with the active ingredient. It gets messy, messy, messy fast. So bees aren't getting exposed to glyphosate in the field. They're getting exposed to Roundup, and there's a big difference. So we're trying these individual things first before we get big and messy, just for that reason. Great question, but it's mainly because my brain's too small to handle the multiple things. I thank you for your attention. You get me 3 more times, so I hope you're okay with that. So thank you, guys.